Hello guys, welcome to my channel SwathiPharmaJnan, sharing knowledge. Today's topic is chemical degradation of penicillins. It contains beta-lactam ring which is attached to thiazolidine ring. To this thiazolidine ring, two methyl groups are attached at second position. One carboxylic acid group is present at third position. Consider this one as the first position, second, third and fourth, fifth, sixth. At sixth position, there is a side chain which is also called as acyl amino side chain. Why? Because this is acyl group or maybe alkyl group or aryl group. At seventh position there is a ketone group. This is the general structure of penicillin. This penicillin when reacts with acid at pH less than 3, in the conditions of pH less than 3, it will convert into penicillinic acid. Penicillinic acid. The whole pinam ring or beta lactam ring with the thiazolidine ring has broken, and it has the penicillinic acid contains oxazole group. Five membered ring with one oxygen and one nitrogen at first and third position is called as oxazole group. To this oxazole group, one unsaturated bond is present, so that's why we can call it as oxazolin. At second position, alkyl R group is present, and at fourth position there is NHCCOOH, CH3, CH3SH. The pinam ring has broken and it has formed into this structure, and then the ketone group is as such at fifth position. This is penicillinic acid. This penicillinic acid, when it undergoes hydrolysis means the reaction when it takes place with water, then this penicillinic acid will convert into penamaldic acid. This part of the penicillinic acid remains the same. The cleavage of the oxazolin ring has takes place, and it has formed into COOH group, and here one ketone group is present and here alkyl group is present as such. So this is structure of penamaldic acid. This penamaldic acid will further degrades into penicillamine. In this penamaldic acid, the main functional group is carboxylic acid. When the penamaldic acid further degrades into another two structures, that is penicillamine and penaldic acid. In penicillamine, amine group is the main functional group. In penaldic acid, carboxylic acid group is main structure. We'll see these two structures which comes from penamaldic acid. Penicillamine contains CH3, CHSH, CH3, means this particular structure is present. The cleavage has been taken place here at this position, and then the penamaldic acid will convert into penicillamine. This particular remaining structure will be converted into penaldic acid. Penaldic acid contains R C double bond O NH CHO COOH. In this penaldic acid, carboxylic acid group is the main functional group. This penaldic acid will further degrades into penicilloaldehyde. What is penicilloaldehyde? Penicilloaldehyde contains aldehyde group as the main functional group. How this penicilloaldehyde will form from the penaldic acid means it will undergo decarboxylation. What is decarboxylation? Removal of carbon dioxide group is called decarboxylation. CO2 group has been removed, so that's why we'll get this penicilloaldehyde. Penicilloaldehyde structure has been formed. Now coming to right part of the penicillin, that this is a general structure of penicillin. Penicillin when reacts with alkali in the presence of penicillinase enzyme, then it will convert into the cleavage of beta lactam ring takes place. Here the cleavage takes place. Thiazolidine ring is as such. Two methyl groups are present at second position. At third position, carboxylic acid group is as such. Only the beta lactam ring cleavage has been taken place, and here COOH group has been formed. NHCOR is remains same. This is penicilloic acid. This penicilloic acid is another degraded product which is obtained from the penicillin when it reacts with alkali or penicillinase. We have completed this particular part till here, and we have completed this degradation product. Now we'll see Penicillinic acid which is obtained from penicillin, when it reacts with acid, that will convert into, this is a reversible reaction. Penicillic acid and penicillinic acid are the reversible product. So when this penicillinic acid further degrades, it will convert into penylic acid. What is the structure of penylic acid? Penylic acid contains thiazolidine ring, and it also contains imidazoline group. At this position, alkyl group or aryl group is present, which is R group, and next here COOH group is penylic acid. This penylic acid further degraded as penyloic acid by undergoing decarboxylation reaction. What is decarboxylation reaction? Same, these two reactions are same. Removal of carbon dioxide. So removal of carbon dioxide takes place at this position. So we'll get this structure, that is penyloic acid. In this structure, there is no imidazoline ring is present in this penyloic structure. It contains thiazolidine ring. Thiazolidine ring, first position, and at second position two methyl groups are present. Third position carboxylic acid group is present. This is structure of penyloic acid. This is the chemical degradation of penicillins. If you practice one or two times, then it will be easier for you to write in the exam. This is one of the important question in medicinal chemistry exams. Degradation of penicillins takes place in alkaline conditions, acidic conditions, or in the presence of enzyme beta lactamase, and in the presence of nucleophiles like water and metal ions. When in the presence of nucleophiles like water, it will undergo the reaction chemical degradation, then that reaction will call it as hydrolysis. Sometimes it may undergo the reaction with metal ions also, and sometimes it will undergo decarboxylation reaction also to produce degraded products of penicillin. Chemical degradation of penicillins gives penicilloic acid, penicillinic acid, penamaldic acid, penylic acid, penyloic acid, penicillamine, penaldic acid, penicilloaldehyde. All these structures are available in this chemical degradation of penicillins chart. This is one of the important question. Practice well. We'll see about cephalosporins in my next video. Thank you.